I'm Sarah Halter. I'm the Executive Director of the Indiana Medical History Museum, and I want to thank you for joining us this evening. And for those of you who tried to join us on Wednesday on Facebook, thank you for your patience and for not giving up. We wanted to be able to stream these events live on Facebook so that more people could watch and participate, and also so that we didn't have to require you to register ahead of time. But that technology is the devil. So here we are on Zoom, our trusted old friend who has never failed us. We are still recording this program, so it'll be available online later on our website and also our YouTube channel. This is a webinar format, so you are all here in listen-only mode. You can see and hear us, at least in theory, but no one can see or hear you. If for some reason you need to use your telephone for sound, you'll find the phone number and password in the chat. And please feel free to use the chat feature during the program to post your questions. We should have some time at the end for a bit of Q&A so that our speaker can address those. Thank you again for joining us tonight. This is an important time for all of us to come together as a community and to support each other. And it's a really important time for all of us to remember and learn from our shared past. It couldn't be more obvious than it is now that the past informs the present and inspires the future. That sentiment is important to us at the Indiana Medical History Museum, and it's been a part of our mission statement for the last 4 years or so. Everything that exists has a history, and there's a historical context to everything that's happening today. Preserving and sharing that past, all of it, the good, the bad, and everything in between, helps us to better understand who we are as individuals and as communities, and how we got to where we are. And acknowledging and confronting that past is the first step toward improving the future. As frustrated as I get sometimes with technology, I do appreciate the opportunities it provides for us to continue to connect with you, even while we can't invite you into the museum's building. That's very important to us, especially right now. And so we're happy to have these new ways to engage with you and to engage you with Indiana's medical history. We're really glad you're here, and we hope you'll keep in touch. We are very active on Facebook and Twitter. We're on Instagram, and I hope you'll visit our website as well, imhm.org. There you can learn about new projects and programs and our preservation efforts, and also check out digital content as new things are posted. So we've been hearing a lot lately about the 1918-1919 flu pandemic and its parallels with what's going on today with COVID-19, and we have a great program about that coming up on July 12th at 2 o'clock. Bill Beck and Dr. William McNeese, both from the Marion County Historical Society, will be joining us right here on Zoom, so I hope you will as well. But the 1918 flu wasn't the only other pandemic we've faced in the last century, excuse me. There's another important one to remember, one with themes that also echo what's happening today, its deadliness, its novelty. It was completely unknown. There was no understanding initially of what it was or how it was spread. It disproportionately affected a marginalized group of people, and as a result, there was a lack of public and government action that might have saved lives. There was a global effort to study it and find a cure, and that work continues to this day. And there's also still a stigma attached to this disease and stereotypes about those afflicted. It's a pandemic that's much more recent. It's still strong in the memories of many of us today, but it's not discussed as openly or as frequently as the flu. I'm talking, of course, about HIV/AIDS. It's a big topic, and admittedly, it deserves a lot more discussion than we can handle here tonight. There are many social and scientific lessons to learn from the AIDS pandemic, but tonight, Dr. William Schneider is with us to talk about the origins of the HIV viruses and the important lessons we can learn from those origins, about where pathogens come from and why some produce epidemics or pandemics and others don't. Dr. Schneider is Professor Emeritus of History and Professor of Medical Humanities and Health Studies at IUPUI, and he's Adjunct Professor of Medical and Molecular Genetics at IU School of Medicine. I'm happy to say that he also serves on the Board of Directors of the Indiana Medical History Museum. His research interests include the history of science and medicine, French history and 20th century Europe, global health history, and humanitarian assistance. His many publications include An Empire for the Masses, The French Popular Image of Africa, 1870 to 1900, Quality and Quantity, The Quest for Biological Regeneration in 20th Century France, and The History of Blood Transfusion in Sub-Saharan Africa. He also edited Rockefeller Philanthropy and Modern Biomedicine, International Initiatives from World War I to the Cold War. And he was co-editor of the translation and annotation of The Story of One Life, the autobiography of the early 20th century Polish immunologist, microbiologist, and hematologist Ludwig Hirschfeld. So welcome, Dr. Schneider, and I will let you take it from here.
Okay. Thank you, Sarah. I hope people can hear me. And it's really an honor to be here and to be able to give a talk. It's a little bit different in that the talks for the Medical History Museum are usually in the museum itself. And of course, the most striking example of the history of the museum is the amphitheater where those talks are held. And I've got a picture of that here so that you, if you're not familiar with it, you can see what it looks like. I think it's important to note that although the museum is in the laboratory, the pathology laboratory of what was Central State Hospital, and its main purpose of the laboratory was for research and clinical purposes, the museum, the building had an amphitheater for teaching, and so teaching from the beginning was important. And in fact, its inauguration in 1896 had a lecture for outsiders with a noted pathologist from Chicago. And so in a sense, the lectures that the museum is giving continue a tradition of 125 years. It's too bad we can't be there, but we'll be there in spirit. Now, the talk for today is the reason we're not there. It has to do with pandemics, as Sarah said, and epidemics. The particular one that I'm going to talk about has to be HIV/AIDS, as she said, which is not the most frequently cited one. I want to say a little bit about a disclaimer as far as my expertise is concerned, because I'm neither a medical doctor nor an epidemiologist. I'm a historian by training, but for the past 10 or 15 years, I've been working on this project about the emergence of HIV, which is the cause of the AIDS pandemic. But it was really a process of me learning from a large group of people that we put together in order to work on this question. About 15 years ago, I was invited by a noted virologist, Preston Marks, and an epidemiologist who were looking at the history of where the HIV virus came from. And they were smart enough, I think, to realize it was a historical question. So they reached out and found a historian that could answer some of the questions that they wanted answered, but I realized I needed help too. And so over the years, we gradually added a team of almost 10 people, which included, in addition to a couple of virologists, an epidemiologist, a couple of medical anthropologists, African historians, and historians of medicine. It turns out to be a complicated question of where these and just how these viruses emerge and can produce epidemics. So I'm going to present the results, a summary of our findings. It's going to be a whole book. It won't take quite that long, but we'll see how I can present it to you in a way that's understandable. So let's start with a question of what a epidemic is. It's, to my understanding, simple enough. I hope the epidemiologist will agree with this description that I have. Let me see if I can get this out of the way. Yeah. It's in a very simple sense, epidemic is a word that's almost overused to mean a rapid increase. In the case of epidemic diseases, though, it's an increase in the number of cases of the disease that over more that's normally expected in a population in a particular area. So increase is important, the dramatic and sudden rise, but a rise above what can be a normally or endemic level of disease, but it also depends on a particular population in a particular area. Pandemic is more widespread, as the graphics on the bottom show you. Now, the reason for these pandemics or epidemics is that either a new pathogen or a change in an existing one that previously was either steady or declining begins to take hold, or it spreads to a new population. And in the case of the AIDS pandemic, it was the result of a new virus, HIV. So as Sarah mentioned, the AIDS pandemic is huge. It's not frequently cited now, but it is not just recent, but it's arguably still going on as a pandemic. So like the definition says, it depends on where you are in the world, as we'll see in a minute. It's also important to study past epidemics. The epidemiologists study it, and people can learn what to expect, what not to expect from epidemics and pandemics that we've had to live through. But I'm going to focus on the emergence of viruses, but initially I'll describe a few of the features of the pandemic and HIV, which are important in understanding the emergence of the viruses. Then describe the most widely held explanation of the emergence and the initial epidemic spread of HIV. And then some of the questions about that we in our research group found about that explanation, and then suggestions we found to, we think, offer a better explanation. So here are some of the basic features of HIV/AIDS. Those of you old enough to remember, this will not be news. Those who don't, lucky you, you didn't live through the initial part. It's a new virus. It is a virus that adapted from Simian virus, meaning chimps, apes, or monkeys in Africa. And the earliest estimate of when this new virus emerged is around 1920. The transmission of HIV is through bodily fluids, but the manner in which it's transmitted can vary in the risk. What we're most familiar with is sexual intercourse, but as you'll note from this CDC graph, it's a relatively low risk. Now, the reason for that has to do with the difficulty of according to what I'm told, difficulty of the virus penetrating the membranes in sexual intercourse. It depends on the viral load as well, but because sexual intercourse is so frequently entered into, the low risk is magnified, and so it's the most common means of spread of HIV over the years, although initially it was a barrier to get over. Unsterile needles we usually take to mean drug addicts via sharing needles, but as it turns out around the world, a much more common use of unsterile needles was in campaigns against diseases, injection campaigns or smallpox campaigns, in which the people giving the injections didn't sufficiently sterilize the needles. And so there are plenty of cases historically of where this happened and spread some diseases. Hepatitis is probably the one that's most frequently cited. But it turns out this is, of course, also the case with HIV. It's a little bit a greater risk. It depends on the viral load, but still relatively low. Much higher risk is the maternal newborn transmission. This is either in utero or through breastfeeding, 25% risk. And this is the only thing that can be said about this as far as epidemic transmission is that it doesn't enter into it because newborns won't be transmitting it to anyone else. Transfusion is by far the most efficient means of transmission, almost 100%. As you can see, the fact that transmission isn't used as much is counterbalanced by the higher risk. And as we'll see later on, transfusion comes in a little bit later, but it was an important and is an important means of conveying the virus. Of course, the case of Ryan White is well known in Indiana, and so it occurred in the US until testing was available. These means of transmission of bodily fluids are less efficient than airborne transmission like the flu viruses or vector transmission via insects, mosquitoes, or other insects, except in the case of transfusion in the maternal newborn transmission. Another key feature of AIDS is the asymptomatic nature of it for as long as 10 years. We are concerned now about the COVID transmission of two weeks of asymptomatic, if not longer. But for 10 years to be asymptomatic after infection and still contagious is one of the main reasons why the virus spread. Even though it was more difficult to spread, it spreads so widely. The way that HIV works is to attack the immune system, which initially is able to suppress it but not eliminate it. And the HIV continues to operate until after about 10 years, it successfully weakens the immune system so that the actual cause of death are the opportunistic infections, pneumonia and rare cancers. And of course, the worst part, of course, about HIV and AIDS is that it is 100% lethal unless it's treated. Now, the way these discoveries were made is relevant both for the current pandemic we're in, but also as far as the emergence. It was in 1981 that the clinical features of AIDS were first noticed and recognized. This took a long time because it's a particularly pernicious way in which the virus doesn't directly attack an organ of the body or an obvious symptomatic feature of how the body works, but it really attacks the immune system. It's only in the later stages, after 10 years, that the symptoms are recognized. It also was complicated because the initial group that was affected were gay men, and that brought with it all sorts of political and social stigma and problems and misleading clues as to what caused the disease until essentially after about a year it was recognized to be a more general virus affecting all people. The actual word AIDS wasn't adopted until 1983. The virus itself, it took 2 years to discover and isolate at the Pasteur Institute. And it was another 2 years before a test was developed for the virus, which was crucial for two things. Most obviously crucial because it meant that one didn't have to wait 10 years for symptoms to be evident of who carried the virus. It could, if one were tested, one could tell right away. And this was important in tracking and tracing and trying to prevent the virus. The other important result of having the test was that it quickly showed up some viruses that were not positive or not negative and in the kind of a gray area. And this fairly quickly, within a year, resulted in the discovery of a second virus, the so-called HIV-2. And it turned out that this virus, which adapted from simians, was from monkey species very far away, 2,000 miles away from the chimp species of the HIV-1. And as people continued to search for both the different viruses that could be genetically sequenced and found to be different, it turned out that there were quite a few different HIVs with separate crossovers from chimpanzees or monkeys. The last feature of discovery that I'm going to mention is the therapy, which took, again, a number of years later. By the end of the 1980s and into the mid-90s, eventually the so-called multi-drug therapy was developed, which could suppress the virus to a level where it was no longer contagious. Of note, which is not on this list, is a vaccine, very relevant for today, not for lack of trying. Enormous time and money has been spent in trying to develop a vaccine for HIV and AIDS, but to this date, it's not been successful. Now, as Sarah said, much has been written about AIDS in film and literature over the 40 years that it's been with us. But I'm going to only show you one thing about it to give you a feel for the scope and the question of what is going on, and it's the curve. A lot of talk about the curve for the current pandemic we're in, and this is the curve for AIDS. And it's, I guess, good news in the sense that at least the curve was bent down, but if you'll notice the amount of time it took to bend was very, very long, and the number of people infected or who died are very, very large. And if you look at the right end, you can see at a peak of 1.7 million deaths. In 2018, there were still 750,000 deaths and 1.7 million new cases. The fact that it's not talked about in the US is that those numbers are lower, but if you look at the numbers, they're still quite high. New cases in the US in 2018, over 37,000 and over 16,000 deaths. I tried to find the curves for the height of the virus, and this is the best I could come up with. And you can see how daunting they are and how similar they look to some of the curves we've been looking at for the current pandemic. The one on the left is the US, and it turns out this was done in 1997, but it's later been adjusted because it's about 1/3 of the actual cases and deaths. At the peak, maybe 40,000 people died in the US of AIDS annually. and 70,000 new cases were developed. In Africa, it was even more daunting, and of course that's where the pandemic has been and is the widest spread. And you can see how in 2003, when this graph was drawn, there was no end in sight. And really, it wasn't until the mid-2000s, 2004, 2005, that the curve in Africa finally bent. So let's turn to the question of the emergence and explanations of the emergence of the viruses that cause AIDS. This is a short list of a few of them. I won't go into great detail, but really, the one at the top is worth noting because most epidemics and pandemics are seen as one-time events, either random mutations of viruses, or if you're a believer, acts of God, or perhaps a CIA conspiracy, and not seen as something that's ongoing and can continue to occur. The scientific explanations have focused on the transmission or the jump from the simians, in this case chimpanzees, to the human virus, and a couple of the explanations that we see here. One is, no time to go into great detail, was the use of chimpanzee kidneys to develop a polio vaccine in Africa that one researcher thought might be the explanation for how this virus that was in chimpanzees spread to humans. It turns out researchers later found that it in fact was not the same chimpanzee that was responsible for the jump, that the HIV virus came from a different species of chimpanzee. The cut hunter explanation is a simple explanation that processing of monkey meat or chimpanzee meat by a hunter who had a cut would become infected. The bottom one is the one we're going to talk more about because it has to do with, it's a little bit more subtle explanation of how a virus adapts from an animal species to a human species. Now, the fact that humans get diseases from animals is not rare. It's quite common. Indeed, the CDC estimates as much as 60% of all the human infectious diseases come from animals. Here are some of the more common diseases we're familiar with and some of the more common animals that we're also very familiar with. Most of these came when humans first domesticated animals about 10,000 years ago, and we've developed the immunity one way or another to the various, or found treatments or eliminated, in the case of smallpox, these diseases. A lesson learned, but the fact is, though, that there continue to be viruses that come from and are discovered from various animals, and this is a long list. It's too small to read. It's several hundred. Going back to 1901, it really is a reflection of the progress of science to be able to discover viruses. Some of these are ones that we're familiar with and that have been around for a long time: yellow fever and rabies, polio virus, and measles. The rest of them are ones that we haven't heard of because they didn't become very widespread. But it also includes, as you'll see in the 1970s and the 1980s, hepatitis virus, which was an old ancient virus that began to spread among humans, Ebola, a virus in bats and other animals that jumped to humans, and then there's HIV. And on this list is the first SARS virus, which also has become epidemic. Now, as it turns out, it's not this jump from animals to humans is not as simple as it's called zoonosis, which means we catch the animal disease. Some are like that, like rabies. We're familiar with rabies. If you're bitten by a dog, you get the rabies virus. But the other ones are more complicated and more complex. Oftentimes, the animal that carries it will infect another animal that will infect a human, and then the human can then infect other humans. So it's spread not just by contact with animals, but contact with humans. The one on the right is the one of most significance because it has to do with HIV. And as we'll see with HIV, the complication is that the animal virus doesn't produce the virus in humans. It had to be adapted to become a truly human virus. So if we look at the discoveries that have been made through the testing and finding of the different human viruses, and also the knowledge and study of the various chimpanzees and monkeys, to match them up. The general scientific agreement from both testing and gene sequencing is that most of the HIV infections are from a human virus that jumped or adapted from a simian virus in chimpanzees in southeastern Cameroon. The most widely repeated estimate of when this first emerged was 1920. This is the methodology for this, as we'll see later, is to construct a genealogical tree, because by genotyping the various viruses over time, we can see which viruses came from which viruses and go back and track to see where the first viruses, when the first viruses occurred and where they were found, to show that they occurred. And in the case of this HIV, it was in Kinshasa, the former Leopoldville of the Belgian Congo, in the current Democratic Republic of Congo. So if we put these three things together, the emergence of a virus in Cameroon and its epidemic spread in Congo, the most widely held explanation is that the Simian Immunodeficiency virus, responsible for AIDS, jumped to HIV in 1920 in southeast Cameroon. It made its way down the river Sangha and the Congo River to Leopoldville or Kinshasa. And in Kinshasa, where there was a higher population concentration, as well as supposedly prostitution and later increased medical campaigns using unsterile equipment, it became epidemic and spread first throughout the Belgian Congo and the Democratic Republic of Congo, and from there worldwide. First through Haiti to North America, and then Europe and the rest of Asia, as well as directly to the rest of the world. So this is the general explanation, and it's pretty straightforward, and it does account for the worst of the virus and the AIDS epidemic. But there's some questions about it that don't satisfy all researchers and didn't satisfy our group, which was doing the research. The first question is that the humans do not catch the simian virus like rabies. And the question is, therefore, why, after tens of thousands of years, since humans have hunted chimps or monkeys and have them as pets, why did it happen in the 20th century? One of the explanations is that there was an increase in hunting, presumably from the introduction of guns. But our research, the medical anthropologists actually studying in Africa, found that there was little use of guns. The colonial authorities didn't really want Africans to have a lot of guns, and nor was there an increase in hunting in the early 20th century. Another question is why there was a delay. If the virus crossed over in 1920 and emerged, why was it not until the 1980s until the AIDS epidemic began to appear? Most important, though, is the multiple HIVs that have been discovered. How and why did multiple HIVs occur? It was obviously not a one-time event. Not all of them became epidemic or pandemic. And so another question related is, why did some of these pathogens become widely spread? So if we return to the question of the multiple HIVs and the different places in West Africa where they developed, we can more clearly see why this is such a question. So of the many species of primates that exist in sub-Saharan Africa, a majority of them, 46 in fact, have been found to have the simian immunodeficiency virus. The fact that so many of them have them means that they are millennial old and they're non-lethal and asymptomatic. So they don't really bother the chimps and the monkeys and the gorillas that have them. And on this long list, as it turns out, only 3 of them had crossovers, but more than one had crossovers that became HIVs. Central African chimpanzees, lowland gorillas, who are also found in the same area of Cameroon, and Sudi-Mangabe monkeys, who are found in West Africa. So here's the Sudi-Mangabe, and here's the West African rainforest where it's found. And here are the various countries where a number of the HIVs of subgroups of HIV2 were found, in fact, 9 of them. This is Cameroon and southeastern Cameroon, where the chimpanzee and the gorillas were found, that produced 4 different subtypes of HIV1. This shows you how the genetic sequencing and genealogical mapping can show how the HIVs are close to the simian viruses. In this case, these are the chimps. Down here, we see the gorillas, and you can see how they were matched and produced these results. So to summarize, we can see that there were 13 so far of these HIV-1 viruses that crossed over. And each one of these, although the ones and the two refer to the different species, the groups represent a different crossover. So a different chimp here or a different monkey produced an HIV that has been found genetically to be different and not a branch from from the same one crossover. Here's a more developed table to show you when the estimate is of the emergence, and they vary from the 1920s to the 1940s and 1950. You can see that this is mostly in Cameroon and the HIV-2s are in West Africa, but you can also see that the epidemic spread was only in the case of 4 of these. The others were in effect what we might call dead-end viruses that didn't affect anyone except the one person that was found, or at most one or about a dozen people. So there's quite a variety. And our conclusion is that not only do we need to explain the different number of jumps, but also that crossover and the epidemic spread have different explanations. So let's take a look at the various epidemics, and you can see this is the one we saw before of HIV-1 group M, which is a jump from the chimpanzees in southeastern Cameroon, and the virus or the chimp or the human made their way down the river to Kinshasa or Leopoldville, where it became epidemic and spread to the rest of the world. As you can see, this is the one that's affected over 90% of people in the world, all over the world, 70 million infections, maybe 30 million deaths to this date. The HIV-2 epidemic occurred in West Africa in the rainforests where the Sudi-Mangabe monkey lives. We found the various HIV-2s in different parts, including Guinea-Bissau, a former Portuguese colony, as well as the Ivory Coast, which is where the rainforest is, and Ghana. And you can see that this HIV-2 did also spread around the world to the former colonies in Portugal, as well as Brazil, the United States. The number of people affected, not as bad as HIV-1 group M, but still 1.5 million people is arguably a pandemic. So those two aside, the fourth, those three HIVs aside, the fourth of the HIVs, group O, was confined to Cameroon and affected anywhere from 20 to 100,000 people. We're not quite sure. This was only discovered after a while when samples that were stored were found to be genetically different, and then the estimates of prevalence showed that tens of thousands. This is clearly an epidemic and not like the dead-end viruses. The dead-end viruses, as we can see, were developed in places, but as I said, there's reasons why these didn't become epidemic, and the main reason is that it was difficult for the virus to spread. But in the cases of four different viruses, they were able to. So what can we find that can explain how 12 viruses jumped to become human viruses, not chance, and how an explanation for this beyond simply going to a capital or an urban center like Leopoldville can explain it. This may have been the case with HIV-1M, but it wasn't the case with the other HIV-1O or the HIV-2A and B. So what made these crossovers possible but rare, and epidemic in even rarer cases? Our conclusion is that the crossover as well as the epidemic was the result of increased human-to-human infections, not a change in the simian-human interaction. So the human-human explanation is best encompassed in a proposed theory called the serial passage theory, and the person probably most associated with this is one of our research group. His name is Preston Marks, and it is part of this more complicated explanation, and I'll try to give a brief account of how it works. It begins with the assumption and the fact that the simian infection from the simian virus is not a simple zoonosis. You don't get the simian virus like rabies when you're bitten by a dog. Within 6 weeks, the immune system of our bodies is able to suppress and eliminate the simian virus and any mutations that the virus might have produced that could last a little longer. Now, in order to test his theory of what happens in serial passage, Marks and a group at the Tulane primate lab did an experiment not with humans to infect them with the simian virus, but they took a human virus, the HIV-2F virus, and infected a pigtail macaque monkey. And what they found in the viral load of the monkey was that the viral load spiked with the HIV as well as variations on it that occur. You get different mutations that occur, but that the immune system of the monkey pretty quickly suppresses the virus in about 6 weeks or so and eliminates it. But then they took plasma from that monkey after only about a week or so and injected it into a second monkey. And what they found, similar to the first, was that the viral load of the second monkey again spiked very quickly, even higher than the first, until the immune system of the monkey was able to suppress it almost as quick, a little bit longer, but it bounced back and bounced back fairly high until it and its variations, as the virus and its mutations tried to overcome the immune system, eventually declined, as in the case of the first. But then they did a third injection of a monkey with the plasma of the second monkey that was injected and injected it into a third one and found again a spike in the viral load and the diminishment. Then it bounced back and then went down, then bounced up and continued on and eventually increased, which means that this monkey had now developed its own virus, which was a mutation, not the same as the human virus, but it could spread between the monkeys. So the conclusion that Marks and his colleagues drew from this, applying it to humans, is if the chimpanzee or the Pseudomangabey monkey infects a human, that human will suppress it and it will not be noticed after about 6 weeks. But within that 6 weeks, if that human passes that virus along to a second person, that person will then suppress it. It'll take a little longer and different adaptations and mutations will arise. And if that is passed a, say, third or perhaps fourth time, then like with the pigtail macaque monkeys, you'll actually have a human virus that can be passed between humans. And this is the explanation of the serial passage for how you can see it's difficult. So on rare occasions, but more than one occasions, the simian virus can be adapted to the dozen or so cases of human viruses. And then some of those becoming epidemic. So the question then is, how could the human-human infection increase in the 20th century? And the answer is colonial rule and things that were developed. That's the most obvious answer. So if we look at the features of colonial rule that have been suggested, they're the usual means of transmission of HIV. We found sexual intercourse, the urban concentration of towns and cities in the colonial rule, and also some of the new medical procedures. Sexual intercourse, we know, is not a good explanation because of the difficulty of passing a partially adapted virus. It just didn't work very well, and it's a very low risk. The urban concentration could have been important as it increases the risk in people that are there, but it's not the case with all three or all four of the virus, all 13 of the viruses that jumped from the simian virus to become human immunodeficiency viruses. But the one thing that was ubiquitous in all of the 13 cases was the use of unsterile needles that were in disease campaigns or the use of blood transfusion with no testing. This is, of course, an otherwise quite laudatory and valuable procedure to save lives, but the unintended consequences of not sufficiently sterilizing the needles or being able to test for HIV and blood transfusion made it possible for humans to pass the simian viruses that could become adapted and become human viruses. And then once they were human viruses, of course, become epidemic. because of the increased ability of passing viruses between humans. So smallpox vaccinations were the first of the most widespread inoculations, going back to 1900 and exceeding 1 million in West Africa. So smallpox had been known in Europe for 100 years since Jenner's discovery, and there were mass campaigns. People were lined up outside the villages, and the various vaccinations were given to people. In the 1920s, in Cameroon and elsewhere, sleeping sickness campaigns again lined people up and gave them injections against people for treatment. There were also anti-venereal disease dispensaries in the Belgian Congo, which were on the order of tens or 20,000 annually, but it was noted by many of the people administering them that it was impossible because of the small number of syringes to sufficiently sterilize them, and they were reused, and contaminated blood caused disease. Same thing in Guinea-Bissau, a colony of only about 50,000 people, 500,000 people. Massive injection campaigns can account for the spread of HIV-2 in Guinea-Bissau. And most importantly, although in the manuals there were various procedures shown for how the syringes should be reused in water and soap and boiling containers, these were obviously not the case given the circumstances of these mass injection campaigns. Blood transfusions, again, it turns out occurred earlier in Africa than anyone imagined, in the 20s and 30s, and then right after World War II in 45, throughout almost all the colonies in Africa, without any screening, and becoming quite widely used, and with the very high chance of passing any viruses between humans. So, to summarize, gone on rather long. The global AIDS pandemic was caused by multiple human immunodeficiency viruses that emerged from different Simian viruses independently in widely spread regions at different times, only in the 20th century. Best explanations we can find for this, both the emergence and the epidemic spread, is the increased ability of humans to pass viruses between them. In order to understand how that occurred, it requires understanding the history and social and cultural circumstances at the time. And what we've concluded is that the most widespread explanation of the ability of humans to pass viruses between themselves that accounts for the HIVs are the unintended consequences of new and otherwise quite beneficial medical procedures, mass campaigns against diseases, and insufficiently sterilized needles and blood transfusion. Broader lessons to conclude for current and future epidemics. Given the fact that these viruses come from animals, people should be guarded and not live in close proximity to animals, especially undomesticated animals. Your dog and cat, we've probably gotten what we can from them, but other more exotic animals, it's probably not a very good idea. Disposable needles should be used for injections because of the difficulty of sterilizing and reusing them. State-of-the-art testing is absolutely necessary for blood transfusions, and especially as new viruses come, there should be tests for them to prevent them being transmitted. Most importantly, any new potential viruses, symptoms, unusual symptoms, must be closely monitored, must be closely and immediately monitored and reported, and above all, this must be done worldwide through the World Health Organization, which has for 50 or more years been for 70 years, the most important organization for checking this. Okay, Sarah, I'm going to turn that over to you now to see if there's any questions, if this came through, I hope.
Does anyone have questions? I don't see any in the chat. I have a couple questions though. Maybe I'll just ask one and then see if anyone has another question. Sure. I was wondering, given that those first cases, where it was first known to emerge in humans, given that that was so long ago and it wasn't widespread and they weren't looking for it, how did you guys determine the dates for those? Was it based on medical records, or do you guys have specimens somewhere that were examined? I'm very curious about that.
For the HIVs? Yes, it's a great story. Could be a good detective story on television. So the way it works is this: the the dates can only be estimated if you have enough of the viruses over a period of time to do the genetic sequencing of them and tell, first of all, which ones came from which and where they are. But also, as it turns out, HIV, as it spreads, has slight variations. And so you can tell from the variations a rate of change. And by seeing the rate of change in the first viruses from the 1980s down to the present time over 30 or 40 years, you can estimate the rate of change and then try to figure out how the viruses that are distant from each other genetically have a common ancestor. And so for the 3 or 4 viruses where there's enough of them to do it, we can estimate within 20 or 30 years, it's a rough estimate because it requires extrapolation, these virologists, not me, have been able to estimate when it occurred. There was a great search when the AIDS first occurred for old stored samples to see if there were some either cases written up or some samples around. And there were some samples from 1959 and 1960 found from the Belgian Congo, from Leopoldville. So we know that those samples had viruses that existed that long ago in the case of HIV-1. In the case of HIV-2, there were stored samples from the 1960s that showed that it was around as far back as then. And so a combination of a few older stored samples and mostly the genetic sequencing and what's called molecular clock estimations. In the case of the 3 or 4 of the epidemic spread viruses, we can tell when.
Thank you. We do have one question from Tom and/or Gail Fenimore. Not sure which posed the question. What do we know about the U.S.'s current dismissing of participation in WHO? I don't know if you can answer that or if you even want to go there, but.
Well, it's important to know that the United States essentially created the World Health Organization in 1948, 47. I've been there and done research there, and it's like an American research center with library. And the U.S. has funded in large measure, and the CDC has largely educated the rest of the world through the World Health Organization. So the United States is very much present in WHO in many ways. The current so-called withdrawal from WHO that has taken place according to our government means that we don't benefit from the knowledge that the rest of the world is gaining about the current virus or any other viruses, nor can we share our experience with them. As far as I know, there's no change on the near horizon until and unless the government changes.
Thank you. Did any of the other attendees have any questions? We do have a couple minutes left if anyone wants to ask a question in the chat. Well, I'm not seeing any.
Either it was completely incomprehensible or it was completely, and no one has a question. Everyone understands it.
No one has ever given a better presentation ever. So thank you for being here. I guess we'll call it a night since no one has any other questions. Thank you, everyone.